Hey, we're back at home and ready to move a little bit further down the path towards contemporary personal genomics and forensic analyses that we have. So let's go ahead and we'll pick up where we left off last time. So, by using the short tandem repeat analyses that we talked about, the FBI has standardized the 13 autosomal short tandem repeats along with the amylogenin sex-linked gene into what is known as CODIS, the Combined DNA Index System. And you've probably heard CODIS by watching CSI and some of the other law enforcement shows, NCIS, CSI, CBU, SVU, I don't know. They all have initials that I don't understand. So this really combines all of these different assays across the entire human genome. And you can see in this picture here, they are pretty much spread with one on not quite every chromosome. Okay, there are only 13, so about half of the chromosomes. And in this database, they include both databases from violent felons, but also from crime scene samples. Now, the FBI database does not engage with every single law enforcement officer. The laws in different states are a little bit different, depending upon how they want to interact with the federal government. But certainly the state of New Jersey, the state of Pennsylvania, and the state of New York, as well as the city of New York, all collaborate with the FBI and share genetic data with them. New York State certainly has the best, one of the best, at any rate, the best forensic DNA analysis lab. They pumped a lot of money into it after 9/11, and in order to identify remains of people who were killed in 9/11, as well. And LA also has, the city of LA also has one of the top forensic units and DNA identification units. The state of New Jersey certainly could be by now in the top 5, certainly in the top 10 in the country. Just tremendous resources have been put into the state police's forensic labs, and they also offer summer internships. So those of you who may have an interest in forensics might be interested in contacting the state. Send me an email if you like. I can give you some web links in order to find them. As of June 2020, the data are not entirely up to date because of the COVID crisis. However, June 2020, there were over 14 million felons' genetic fingerprints in the CODIS system and 1 million crime scene profiles. Interestingly, about half of those profiles actually match some of the felons that are some of those 14 million felons that are already in the database. So the CODIS system is very powerful. I like to think of it as the mystical power of CODIS. Certainly, if you watch the TV and you watch movies, everybody says this is a tremendous tool. It's amazing. We can identify suspects if only we have a little bit of DNA. And maybe if you like bad Tom Cruise movies, it can prevent as well as solve crimes. Actually, the Tom Cruise movie was from a great science fiction short story by Philip K. Dick. I highly recommend the story, not so much the movie. There are, however, other specialized PCR-based systems to identify individuals. One of them is mitochondrial DNA analysis. The other is the use of short tandem repeats, but those that are specific to the Y chromosome. And something that is clearly extremely important, we're working our way towards, is single or sometimes called simple nucleotide polymorphisms, SNPs, which we say SNPs. First off, there's mitochondrial DNA analysis. Notice if you were to take a peek at the mitochondrial chromosome, you know that there are 2 organelles in eukaryotic cells that contain their own DNA. One of them is the mitochondrial DNA. The other, of course, is... This could be a quiz. Well, if you don't know it, look it up. Observe how similar the circular haploid chromosome in mitochondrial DNA appears to the bacterial chromosome. And you can see it's rather small. It's only about 17 kilobases or 17,000 base pairs. Okay, so 17 kilobases compared to about the 3.5 gigabases that are found in the human genome. Mitochondrial DNA is great. It has what we call single cell sensitivity because every cell contains 1,000 mitochondria. One of my little pet peeves about the drawings of the cell that you very often see in the beginning of biology textbooks might have 1 or 2 mitochondria in it. In reality, nothing can be further from the truth, okay? How can 1 or 2 mitochondria actually become the powerhouse of the cell? It's not like it's a giant little atomic factory, okay? It's not. There are thousands of mitochondria. Some cells actually have more than 1,000 and have closer to 2,000 or 3,000 mitochondria in each cell type. Great because it's especially useful for shed hairs, burnt remains where the cell or the DNA, the nuclear DNA may be damaged. Very handy. And of course, it can be used to establish relationships because the entire portion of mitochondrial DNA is maternally inherited. Again, recall the slide sets on mitochondrial DNA and maternal genetic inheritance. Really, 97 to 98% of the mitochondrial complement is derived from mom. The other 2% or so can be found from the dad, but very rarely is there a sufficient amount to create a problem for mitochondrial DNA analysis. The problem with hair is a very interesting one, and here are a couple of different types of graphics of the hair. Of course, we have our drawing of hair, a little cartoon, and alternatively, we also have a couple of different types of hair. The first is actually a growing hair, so you could see the root here and the bulb of hair. Sorry about that. And then, of course, this hair, notice that the bulb is actually, or the root of the hair, is actually very small and ovoid in shape. And that's because this is actually a shed hair. So this hair, first hair, was actually pulled. This one is shed. The advantage, of course, of these is that we see that down here in the bulb, in each of our pictures, there are multiple cells. We see there are even some blood cells because of the blood supply to the hair follicle itself. And so, of course, there's a large amount of nuclear DNA to be found, as well as mitochondria. Of course, those of you who've been in the barber shop know that when you cut your hair, there are no cells. It is just protein. And in fact, for a long time, I would make fun of CSI analyses where they would collect hair in a barbershop and say, aha, there is the DNA, and we can match the killer to we're using the COVID database to the crime scene, and they catch the killer. And I'd always make fun because I'd say there is no nuclear DNA. But curiously, what does happen as hair grows is as the hair grows out of the shaft, what you can't see here are mitochondria actually stick to the hair. And as the hair grows, the mitochondria also come off with it. And so hair that is very, very close to the scalp actually does have genetic material. Now, not nuclear DNA, but certainly has mitochondria and mitochondrial DNA attached to the hair. The more you wash your hair, obviously the mitochondria fall off in the presence of soap. But if very, very close to the scalp, there are actually mitochondria, and sometimes cut hairs can be a source of mitochondrial DNA that can be used for identification. So you have to be a little bit careful with the idea of DNA collected from hair. The downside of mitochondria, of course, is also because it has single cell sensitivity, the possibility of contamination is very high. It is possible some individuals, just by chance, have a higher concentration of paternal mitochondrial DNA in some tissues than in others. So this is called heteroplasmy, and this is a problem that can occur. And of course, there's a lower power of discrimination compared to nuclear DNA because maternal relatives all share the same mitochondrial DNA. So I have to be a little bit careful, a little bit cautious. This was a picture taken from Nature magazine 2 years ago, and this is actually the sampling of a single mitochondrion, from a nerve cell on a microscope slide. So really quite a spectacular technological feat where they actually took a mitochondrion from a single cell and were able to analyze the DNA in the mitochondria from a true single mitochondrion. So really an exciting kind of experiment. Are these things done routinely? No. These are very difficult experiments, but this one was proof of principle that it actually could be done. Another one, of course, is the short terminal repeats found on the Y chromosomes, and this makes sense from a forensic analysis point of view. 99% of all violent crimes are committed by men. So why bother focusing on nuclear DNA, which includes females? Let's just focus on the Y chromosome and catch the bad men. And this becomes further because many of these violent crimes committed by men are men assaulting women. Many times a mixture or contamination of a male and female suspects' biologicals pose a problem for analyzing DNA. So by focusing only on the Y chromosome, assuming that there's not a chromosomal abnormality in the person who was attacked, you could actually focus more clearly on the individual. Here is a classic map of the Y chromosome. Notice at the very top is our buddy testis determining factor. A couple of my favorites, of course, are the spitting gene known as the Pituitary gene. Unfortunately, I do have an anti-stop codon, which you may be familiar with. Okay, it's a joke. A good friend of mine from my first postdoc at Columbia made this. You could tell this is a copy of the original that she did on an old Macintosh computer. Here's what the Y chromosome really looks like. Notice that the Y chromosome has known genes only existing to here, and there is very little known about the distal Q arm of the chromosome. And in fact, the Q arm has been shown of late to actually becoming shorter and shorter and shorter across thousands of years. And really, so there is at least one theory that suggests at some point in time the Y chromosome will just be a simple microchromosome with perhaps not even a centromere. From our genomic database, one of the things you could see is how many variations have been identified, and you can see there's a considerable amount of variation surrounding the centromere in the Y chromosome. This really leads us to focus on single nucleotide polymorphisms, or SNPs. SNPs are point mutations, just simple single base substitutions that are found in 1% or more of the population. So there's a population genetic aspect to a SNP. It is just not an individual mutation in a single person or a single organism, if these are used in other species as well. And each person has about 4 to 5 million of these SNPs within their genome. Generally, and what this is a picture of here, is a microarray plate with fluorescent tags, which is each of the individual dots is an individual SNP. And the current chips that are being used, by the way, I made this the size of a slide just so you could see it. These are actually smaller than a microscope slide when they're actually looked at by the computer. There are others of these that are not just visual, but are actually integrated circuit chips that interact with DNA. So there are a lot of really exciting brand new technologies, but this is a pretty picture, and it gives you an idea of how many assays can be done at the same time on a single DNA sample. So this is really quite fascinating. I should indicate that the current chips that they're using, for example, if you send your DNA in to 23andMe or Ancestry.com or MyDNA Heritage, you will get the results for approximately 750,000 of the 4 to 5 million SNPs that exist within your genome, and we'll talk about that just a bit. These SNPs are so unique, thinking about this for a moment, if we can identify 750,000 individual SNPs within your genome, we can use the pattern of those SNPs for identifications, for forensics. We could use them for gene mapping, for genome-wide association studies. We can use them to predict disease susceptibility. Actually, the United Kingdom, in fact, much of the European Union are using DNA identification to track immigration and citizenship within the Union. We do not do that in the United States, but my guess is the time may come where we will be. SNPs are used to predict specific genetic traits. Sometimes a SNP can be found within a coding region, not necessarily, but it could be found within a coding region. And of course, SNPs can be used for classifying patients in clinical trials. One of my consulting clients is doing a long-term series of studies on a disease, which I have a non-disclosure agreement. I can't tell you the company. I can't tell you the disease. But I will tell you it's a very sad disease, and one in which very often there are multiple medications that can be used to treat this disease. And if one is diagnosed with this disease, very often it takes multiple prescriptions to find out which of these drugs is the best one to treat that particular disease for that particular person. Now, wouldn't it be great, And oh, by the way, it takes a month or longer to find out if the drug works. Well, wouldn't it be great if we were able to take a DNA sample and directly identify the type of drug that would be appropriate so that a patient would not have to live through 6 or more months of trying different drugs only to find the one that works. So it can be used for classifying patients in clinical trials. So this is a really hot topic now in pharmaceutical companies. Approximately 50 SNPs provides the same power of discrimination as the CODIS database. So 50, and how many do we actually measure? 750,000. Okay. And so you could see the value of SNPs in the rather than the COVID analysis to identify individuals and to utilize for forensic analysis. Unfortunately, because there are 14 million individuals already in the database and many of those original samples have been lost, it would become extremely difficult to convert the system, which is based on 13 short tandem repeats, to 50 and certainly 700,000 individual SNPs. So these SNPs can be used as predictors of ethnicity by private sector labs. There are a handful of them, and I'm sure you know. And there is a bit of a flawed logic in the use of SNPs for ancestry. First of all, the SNP profile could be inconsistent with the subject's reported ancestry. I have a friend who thought that their mother's name was Waldis from Sweden. And when their DNA profile came back, they came back as predominantly Hispanic. Well, how did the Swedes have Hispanic ancestry? And it turned out that the great-grandmother's name was not Waldis, it was Valdez, and they moved from Spain to Sweden. And the Swedes could not pronounce Valdez, so it became Waldis. Well, guess what? You turn out that perhaps the family history could be inaccurate. Another one is perhaps the SNP profile is inconsistent with the subject's physical appearance, okay? So then they say, oh yeah, well, it's just a matter of human migration. So if somebody is very dark and lives in a place where there are a lot of light-skinned people, they say, oh, well, they must have showed up by human migration. So the trouble is SNPs can be a little bit all too powerful because they can be used for multiple types of explanation. A great example, of course, was from the LAPD, a detective in their forensics unit. The SNPs say he is 62% European, but his appearance is that he is Southeast Asian or Indian. Okay? I like to always ask the question, what about me? You guys have been looking at my face for quite some time now in these videos. And actually in class, what I would tend to do is if we were face-to-face, I would pass everybody out a little piece of paper, and I'd ask you to write down what you think my ancestry is. One of these days, I'm going to figure out how to put a quiz in here to actually get that going. But what do you think? Take a second. Take a guess. I would then collect all of the cards and then summarize them, and in the next lecture, show you, according to 23andMe, this is me. My genes come from mostly Europe, Now, to give you a little bit of family background, all four of my grandparents came from the same little town in the coast of Italy, migrated to the United States in a variety of years, ranging from the early 19 teens to the 1920s, met, and of course ended up having my parents born in the United States, and I was born here as well. So, but look at the tremendous range where perhaps all of my genes have come from, okay? We have a huge range. Now, looking at the geographic range, you might say, well, hey, that's kind of interesting. But it really doesn't tell us about the percentage of genes. Where are the bulk of my genes from? And it's kind of fun to look at a more detailed analysis. It turns out, of course, that I am about 88% European, the bulk of which is about 73% Italian, and mostly from the Campania region of Italy, which is correct. That's where my grandparents hailed from. But notice that there's a little bit of everybody else in here. There's a little bit of everybody else, which makes sense because there's probably shared genes from a lot of geographic areas. What was surprising was looking at nearly 11% Asian, certainly Western Asia, and in particular North Africa. And you can kind of take a peek down here. You could see that about 6% of my genes came from Africa. So perhaps explaining some of the darkness in my skin color, how I tend to tan very rapidly in the sun, my dark hair as well, even though it's turning a little gray now and again. You guys can see that all too well, I'm afraid. So this is something that we see in a little bit more detail, and if any of you have done the 23andMe or ancestry.com or My DNA Heritage or DNA Witness or any of these companies that do this, this is how they determine it, by analyzing SNPs. They compare your pattern of single nucleotide polymorphisms with patterns of polymorphisms that are found from families around the world. Okay? In order to have a reference population, they actually take families that have lived in a particular geographic area for 6 generations. So they became the de facto standard to compare your genes to genes from other geographic areas. So this is quite a bit, it's a lot of fun, but I do want to tell you this is for entertainment purposes only. Entertainment purposes only. Why? Because if you go back, I have some friends when 23andMe was first getting started. I have some friends and colleagues who are on their scientific board, and I was one of the first 1,000 genomes that were analyzed by 23andMe. And at the time, I showed up as being 75% European, and in particular European Italian, which correlates rather nicely with that 73%. But I was also identified as being 25% African. Over the years, that 25% has fluctuated up and down and around, and now it actually got down as low as 2% North African, and now it seems to have gone up a little bit to 6.3. So the prediction of your ancestral combination of genes is very much dependent upon who they are comparing your genetic complement with. So you have to be very careful, because as the reference population changes, so do the statistical analyses of your particular genome. So something to keep in mind if you're looking at those results. So again, entertainment purposes only. A couple of SNP flaws: privacy issues. SNPs can be correlated with a lot of disease states. They can be correlated with quite a bit of personal information, so they would certainly they could possibly violate HIPAA regulations, and for better or for worse, SNPs require a large quantity of high-quality DNA, as you might imagine. So SNPs can be used, we talked a little bit before, for physical mapping, functional analysis, these genome-wide association studies. Really interesting work being done with the evolutionary studies. And I think it's important to recognize that in a lot of areas of science, null results are extremely important, and they have a tendency to become underreported. It is equally important for geneticists not only to focus on areas of variation, polymorphisms, differences in the types of gene sequences, extremely important. But what is equally important are invariant areas of the genome. And so in order to study them, we have to be in a position to review null results as well as positive results. So that really brings us to the end of this mini lecture. I hope that you've learned a little bit more about the state of the art. I'd like to spend a little bit of time in the next lecture just talking about the mechanisms by which we actually identify these SNPs. While many are identified in the manner we talked about before on small chips, there's quite a bit being done right now with what is called next generation sequencing. And we're going to see that these are very, very powerful, very fast techniques for analyzing DNA from both people as well as other organisms. You guys take care, stay safe, see you next time.